All right, so I'm going to take 15 minutes and teach you nomenclature. I'm not really going to introduce lipid biology because that would take too long. But what I want to do is make sure that when we talk about lipids, or when TM talks about lipids and Tanu talks about lipids, we're going to be all on the same page. And then just, some fun facts here and there. So biological molecules in human plasma. Gary Patty admitted to me last night at dinner that when he came up with 1,500 metabolites, this was a subset of metabolites. And so his real space is a little bit different than I think he implied, and he apologized. But it turns out that we're measuring a subset of metabolites simply because we're not measuring, at least at this point, all the potential lipids. And I'm sure all of you know that lipids are a very complex sort of chemical class of compounds. A lipid is really, hydrophobic or amphithetic small molecules, by carb and ion-based condensation of thiol esters, which, really is, what lipids are. I sort of like Orson Welles, when he's talking about, eating, and what he eats is, my doctor told me to stop having intimate dinners for 4 unless there are 3 other people, which I always liked. I tried to find a pithy quote about, eating fats, but I couldn't find one, so this is as close as I could. So really, it's as you all know, that lipids come in a lot of flavors. And if you want a really good sort of overview and understand a lot of this kind of stuff, the UC Davis Lipid Blast project has a lot of stuff, and I've appended at the bottom of the back end of the slide set that I uploaded. There's a lot of structures, so you can actually take a look at them and what are sort of these supergroups of lipids are. But I just want to briefly go over it. So in the distribution of lipids in plasma, which is a lot of stuff that we measure, is that the sterile lipids are the biggest class of lipids out there. So cholesterol and cholesterol derivatives are really the bulk of the hydrophobic lipids, and it's probably the ones that we actually measure the least well right now. We can measure the cholesterol esters fairly well, but the number of different steroids that you can make is huge, and measuring them is something we do, but we have a hard time measuring certain classes accurately and reproducibly. Glycerol lipids and glycerol phospholipids are obviously large classes. Glycerol phospholipids make up all our membranes, and then some of the other smaller classes. Okay, so if you look in Lipid Blast, which is this compendium of all these molecules, you can see that there are at least 29 different classes of lipids that are out there, and the names of these things. So when you actually go through and you start reading papers, you'll obviously see PIs and PGs and CLs and things like that. But some of the ones at the bottom of the list are even strange to me, and I've been doing this for a while. And so I think, so TM, we measure about 15 different classes now, or plus or minus. It depends on how you parse data, and TM will go into that. So here's the type of lipids. There's fatty acids and fatty acid derivatives, including prostaglandins, wax esters, and things like that are generated from most of the prostaglandins and other eicosanoid products are really derived from the polyunsaturated fatty acids that we consume. Glycerol lipids we'll go through, but those are diacylglycerols, triglycerides, things like that. Glycerolipids, single lipids. And so what's a fatty acid? This is elementary biology, and what an ester group is. But this really does give the basis of most of the lipidology that we'll talk about today is really the fatty acids and the kinds of fatty acids that are stuck to the specific hep groups that define the classes. Monounsaturated fatty acids versus like oleic acid, a very common lipid in the lipidome, and then polyunsaturated fatty acids. And most of you probably know that omega-6 fatty acids have the first double bond at the 6 carbons in from the tail end. So this is the omega, which is the last carbon, and so 6 carbons in is omega-6s, and then if you go in 3 carbons in, it's omega-3 fatty acids, and this is really how the omega-3 and omega-6s that you hear about all the time are really defined. And these get stuck onto other things and are have their own biology. So triglyceride, 3 on a glycerol backbone, pretty simple. And then, and the reason that these actually become fun to play with, as it were, under different conditions is that these turn over fairly rapidly. As I showed earlier when we were doing the feeding studies, this is the group that actually changes the most robustly and the quickest. And probably because if you take one lipid off of a glycerol backbone and put another one on, it will change a class and it will change the lipid. And so there's going to be inverse correlations when you actually turn these lipids over, especially in the presence of other fatty acids. And so it's you can play a game in a way to of how things come in and come out of these different lipid subclasses. So the glycerol phospholipids have a number of lipids. So all phosphatidylcholine, phosphatidylethanolamin, the primary lipids that are in membranes. Phosphatidylserine is there. They undergo specific hydrolysis by specific subsets of enzymes generating specific signaling lipids. And the same thing for phosphatidylglycerol, phosphatidylinositol, which is important for a lot of membrane signaling events, and phosphatidic acid, which is does not have the head group, but it just has the phosphate on it, but is an intermediate in the triglyceride synthesis pathway. Plasmalogens are a class of compounds that instead of having a carboxy group, there's actually a double bond in the aliphatic chain that's on the end. The biology of these are not as worked out as a lot of the other classes, but we can identify these in our data set. If they change, don't come to me to ask me what it means. But other people do, and then there are some interesting biology around these. The sphingolipids, which are turning out to be pretty interesting signaling molecules themselves, both through generation intracellular events, but also binding to membrane receptors. Sphingosine, you all, I'm sure, heard of ceramides, which are considered molecules that are associated with triggering insulin resistance, apoptosis, and things like that. These are, bad lipids, but they really are a fatty acid, sphingosine with a fatty acid on there. And we have these are the structures. You can see it's the fatty acid that has an amino head, and then if you take another fatty acid, usually a saturated fatty acid that on the amino group, that's when you make a ceramide. And the sphingomyelins have an additional choline molecule on the phosphate. So giving these variety of lipids that have different again chemical properties, that have signaling properties, and lots of different biological events have been ascribed to these different compounds. And I'm not obviously I don't have time to go into all these. As I indicated, the steroid steroid-derived molecules are the most common lipids that we see in the plasma, at least, and This is a cholesterol ester, and the acyl-CoA cholesterol acyl transferase generates these molecules. And this is one way that a lot of cholesterol circulates around in the blood. And then the hydrolysis of that ester bond to take the fatty acid off delivers cholesterol to the membranes. Obviously, bile acids and bile salts, which we measure, and Casey Thoreau up there has been measuring a lot of these and showing some really interesting biological actions of these compounds. There is emerging evidence that high levels of bile acid in the blood may be insulin sensitizing. There's now repeated reports that Roux-en-Y gastric bypass is associated with high bile acids and bile acid derivatives in the blood, and that may be part of this long-term improvement in metabolism that people see after gastric bypass. Now, whether this is related to decrease in the recidivism of weight loss is also being investigated. There's evidence for and evidence against bile acids playing a role. But we do know that when people get really high bile acids in their blood, they don't get hungry, but they're usually pretty sick anyways. This is a chart. I just put it up here just so you have a reference when you want to start looking at the different particles that we carry around the blood. So if you take blood and you just extract the lipids out of blood, you're taking these lipids for the most part off of carrier proteins. So, lipids are, fatty acids are bound to albumin. That's the only way they can go around the blood. Otherwise, you've got soap, and soap is not good in your blood. Most of the other lipids circulate around bound to protein particles, and the protein particles themselves are differentially enriched in different kinds of lipid species. And so if you find lots of phosphatidylcholines high in your blood, it's likely due to either the HDL particles or VLDL particles. And so you can actually make some guesses about if things are going up and down, why they might be going up and down. You can do secondary experiments related to that. So this is a good reference for you. And then all this stuff afterwards, actually this slide here should be before this slide. But it goes through, and you can just take a look at all the complex structures that lipids can make, and just gives you an idea of, when you see something like, when you see a name of a compound, you can at least go back and take a look at what does this really mean. Because, I think besides myself, Tanu, and TM, none of us remembers well, actually, I don't even remember all these structures. But they are, again, enriched in different places in the cell. For instance, cardiolipins are highly enriched in mitochondrial membranes. And so if you see changes in extracted tissue and you see changes in cardiolipins, it usually means there's probably some change in mitochondrial structure or function, or could be related to mitochondrial structure and function. And these are the inferences that you need to derive from the biological knowledge that you will attain once you do the lipidomics platform.